بسم الله الرحمن الرحيم. طلبتنا الأعزاء، كما وعدناكم بتحميل محاضرات فيديوية. إن شاء الله اليوم راح نحمل فيديو حول Principles of Polymerase Chain Reaction لطلبة المرحلة الثانية. The objectives of this lecture are the following. First, to have an idea what's PCR and what are the uses of this technique. Also to highlight the advantages of this technique over molecular cloning. Number 3, to know the general applications of this method. And fourth, to show how this procedure is used to help diagnosing a case of cystic fibrosis as an example for inherited disorders. So what's PCR first of all? Actually, the polymerase chain reaction, or what's known as PCR, is a test tube method used to amplify a certain target DNA sequence. That's to say, I have a certain piece of DNA which is interesting to me. I want to do some analysis over this piece of DNA, so I want to amplify it. And the amplification here doesn't mean to increase the size of this piece, but actually to increase the number of the copies of this piece of DNA without any cloning indeed. So what are the main advantages of PCR over cloning? As you remember in the last lecture, we have talked about the cloning. So now what's the difference between the two? Or let's say, what's the advantages of PCR over cloning? First of all, the PCR permits the synthesis of millions of copies of DNA. Actually, it reaches up to 1 billion copies of DNA within a very short time. It's just a matter of few hours. Actually, 3 or 4 hours may be enough. While if we compare this to the cloning takes few days. Second point is that the polymerase chain reaction can amplify DNA sequences whatever the initial concentration of that particular DNA is. That's to say, it is very highly sensitive technique. Even if the concentration of DNA is initially 1 part per million in the initial mixture, it can amplify this sequence. Number 3 is that this method can identify whatever DNA is. Whether this DNA was bacterial, viral, plant, or animal, it can amplify it simply and in a very short time actually. Now we want to know what are the steps of PCRs. So the first step is going to be the denaturation. So the first thing is to denature DNA. What does this mean? It means also melting of DNA. That's to say, to separate the 2 strands of DNA to make them 2 single-stranded molecules rather than double-stranded to start with. So as this picture shows to you, initially the DNA was a double-stranded molecule. This is known as a double helix. And then by heating this molecule up to 95 degrees centigrade or so, the 2 strands will separate into 2 different single strands of DNA, so that each of them is going to be as a template to synthesize a new complementary strand. And in this way, we are going to multiply the molecules of DNA in an exponential way. After separating the 2 strands comes the next step, which is the annealing of primers to the single-stranded DNA. So primers in Arabic, it means al-badi'at. So these primers are used to help the enzyme in the next step to synthesize the new strands. So the primers actually are short, and when I say short, they are actually from 18 to 35 bases or nucleotides synthesized in order to attach or anneal, attachment or annealing to the template or to the 2 single strands of DNA that have been produced by the first step. So in order to synthesize and to design the primers, I have to know what's known as the flanking sequences or the flanking regions. So the flanking regions means those sequences that embrace the target DNA. So you have the target or the interesting piece of DNA, and then you have to know the sequence to the right and to the left of this path. These are the right and the left flanks. So these flanking areas or these areas surrounding the target, once you know the sequence of these areas, they will form templates to synthesize 2 single-stranded oligonucleotides or stretches of DNA. Maximum length is going to be 35, and actually they are between 18 and 35 bases long. these are going to be complementary to the respective flanking areas. And these oligonucleotides are going to function like the primers for the synthesis of the DNA later on. So now this is the first step, which is the denaturation. The double-stranded has been opened up and two single-stranded have been now separating from each other. Later on, each of these two initial single strands will work like a template to synthesize a new molecule or a daughter strand. But first of all, we have to know what's the target region. This is the target region. I mean, this is the piece of DNA which is interesting to me. I want to amplify it. Then, because you know now this is the target, this is the flank area, and this is another flank area. Both of them are surrounding the target. If you know the sequence of this piece and this piece, then you can design the primers. This is the first, this is the second primer. These are short pieces of DNA, single strands again, and then they're going to anneal or to attach to the template in a complementary way, meaning that if you have in this flanking area adenine, then in the primer there will be thymine. If you have here guanine, then there will be cytosine. In this way, there will be hydrogen bonding between the two strands, and this is going to act like the annealing force. So you have now two primers, one per each initial strand. Now, because the primer is a piece of DNA, so it has 5' and 3' end. Always the 3' end is facing the target area. As you can see, this is the direction of the arrow, illustrating that this primer, this is the 3' end of the primer, is facing the target. Also for the other primer, the next strand, also the arrow is going from the 3' end towards the target region. And as I said, that the annealing means attachment. After that, the chain extension will start up, and the extension means that there will be a DNA polymerase enzyme working to add deoxyribonucleoside triphosphates or nucleotides. And actually these nucleotides will be added in the experiment in very large amounts so that the enzyme can utilize them and that the process will continue without any stop. So these nucleotides will be added to the 3' end of the primer. And actually at the end of the first PCR cycle, the initial double-stranded DNA molecule will become true. Okay, so this is the first step, is the denaturation. After that, the primers come up here. We have forward and reverse primers. The 3' end of each primer is facing or directing itself toward the target area. Then those with red are new nucleotides being added by the DNA polymerase enzyme. They are going to be added continuously like that. And this attachment is going to be in a complementary way. So by the end of the first cycle of this PCR, you can find that the first double helix become 2. So this is the duplication actually of the DNA molecule by PCR. And after the first cycle ends up, then reaction will be repeated. I mean that the mixture is going to be heated again. There will be melting or denaturation of the DNA. There will be some cooling down so that allowing the primers to anneal or to attach to the two strands of DNA. And then the process will be then the elongation further and further. Then the cycles will be repeated so many times. Actually, the maximum number of cycles will be like 30-35, and usually they range between 20 and 30 cycles. And there will be in each time there will be further increase in the number in an exponential way. That's why we call it as polymerase chain reaction. There will be a chain-like increment in the number of copies of DNA. This figure shows that this is a cycle of PCR. Each cycle continues of heating to start with for a certain time, like in this example is for 1 minute at 95 degrees centigrade. Then after that, we cool the mixture down like to 60 degrees, for example. Also for a given time, like also 1 minute. This is just an example. It's not mandated to be 1 minute. And then this is the time and the temperature allowing the primers to anneal to the DNA. After that, we will go and raise the mixture temperature like to 70 degrees or 72, for example, also for certain time. And this is the phase of actually extending the primers or elongating the primers. So this is a cycle. This cycle will be repeated, as I said, about 30 times. So by the end of this, there will be what? There will be a further increase and increase and increase in the number of DNA to reach up to 1 billion copies. Who is responsible for the increase in the DNA? As I said, that there will be an enzyme called Taq polymerase enzyme that acts to add a nucleotide to the 3' end of the primer. Now, this Taq polymerase is called Taq because it's taken from a bacterium known as Thermus aquaticus. And this kind of bacterium actually has been taken from I think from hot waterfalls where they can tolerate the high temperatures. So the use of their Taq polymerase enzyme can ensure that this enzyme will tolerate the very high limit of temperature during the reaction. As I said, it reaches up to 95 or 96 degrees centigrade, so it can tolerate this high temperature, and that's why this enzyme is going to be added only one time, actually at the beginning of the experiment. There is no need to add it by every cycle. Why? Because it can be added only from the start. It can tolerate the high temperature, it can persist working, and so on. Now, what are the applications of PCR? Why we use PCR? In which cases we use the PCR? First thing, in genotyping. So this means that I can use PCR to know what is your genotype. And we use this technique actually for really amplifying the particular suspicious DNA, because I suspect somebody to have certain disease, certain mutation is there. So I have to amplify his DNA first of all, because I cannot work on only single copy of DNA. I amplify that particular DNA by PCR, and then I go for the next step, which is going to be sequencing, for instance. So the first thing is to amplify DNA and preparing this DNA sample for sequencing, to know what is the genotype, to know whether there is a mutation or not. Second thing is for early diagnosis of serious infections can be caused by low abundance in nucleic acids. Now, if you have heard about HIV, and even nowadays there is the coronavirus, these viruses, when they enter the body, they go through a long or relatively long actually latency period. There will be no symptoms or signs, there will be no clinical manifestations for weeks or for months. So that we know, because these viruses are of this character, we want to diagnose them as early as possible. So once you have only a little amount of the genetic material of the virus, okay, so you can apply this sensitive method to amplify the viral DNA and to say that yes, there is a viral DNA or there is no viral DNA based on the amplification technique. The third use is the forensic analysis of DNA samples. Now, this is used for actually detecting the criminal subjects in cases of crimes. Once you have a piece of the suspected criminal subject, pieces of his hair or a little blood spot, then you can use PCR to amplify the particular DNA at the place of the crime, and then you can return this DNA to the one of the suspected people. And actually this relies on detecting what's known as the short tandem repeat polymorphisms, or variable number tandem repeats. Actually, these are of different types, but we call them as microsatellite or mini satellites. And by that we mean the satellite is a tract of actually repeated sequence of DNA. For example, we have like AG, and this will be repeated like 5 to 50 times in different people. And this pattern of repetition never repeats in another subject. So by analyzing the piece of hair with the blood of the suspected criminal subject, we can say that this DNA belongs to that person or not. Also on this base, we can use actually DNA also for paternity testing. Number 4, for carrier detection and prenatal diagnosis of some inherited diseases. And in this lecture, I'm going to talk about cystic fibrosis as an example for those diseases that we can diagnose by PCR. And cystic fibrosis is actually a disease that affects many systems in the body, mainly the respiratory, GIT, and also many other systems. And it is due to a mutation actually in the cystic fibrosis transmembrane conductance regulator gene. It's known as CFTR gene. This gene is present in chromosome number 7. Q means in the long arm of this chromosome, because as you know, every chromosome is composed of long and short arm. Then this gene is present in the long arm of a chromosome number 7, in the locus or in the region number 3, band number 1, subband number 2. And this gene is almost like 230,000 bases and containing around 27 exons. This picture shows the chromosome number 7. So this is the short arm, which is abbreviated like P, and this is the long arm, which is Q. So in this position lies the gene CFTR with the red color. If we amplify that piece, then we can find that this gene occupies like 230,000 bases. It starts from the position 117 million something to 117 something as well, but the region is like 230,000 gene in chromosome number 7 in the region I showed to you already. Now, this gene actually codes for the production of protein known as CFTR protein, which is composed by around 1,480 amino acids, or 1,500 approximately. And this protein works like a chloride channel. So it is responsible for helping how water, sodium, and chloride passes through the cell membrane, and mainly in the epithelial tissues. It also helps in production of sweats through the skin, also of digestive secretions of the pancreas, and also the mucus in the respiratory passages, mainly the lung and bronchi. So mutations in this gene will lead to change the characters of this protein, and then ultimately ends up with cystic fibrosis. And if we return to the statistics actually around the world, they mentioned that there are almost 1,500 mutations that can cause this disease. But actually the most common of these mutations in cystic fibrosis transmembrane conductance regulator gene is what's known as delta F508. Delta refers to the deletion, F refers to phenylalanine, and then 508 refers to the position of the lost amino acid. So actually, as I said, that the protein is around 1,500 amino acids, and as you know that the protein has an amino acid site and then or amino site and the carboxy terminal. So if we start calculating the amino acids from the amino side towards the carboxy side, if you stop at the area 508, so at the 508th position, normally there should be phenylalanine. Now a mutation is going to happen in the gene so that there will be loss of 3 nucleotides. I mean in one codon, because as you know that every codon codes for one particular amino acid. So there will be loss of 3 nucleotides in the gene, which is reflected as loss of phenylalanine. So there will be deletion of, as I said, phenylalanine from its position, which is 508. So it's only loss of one amino acid, but this loss of one amino acid can lead to impaired and defective folding of this protein and loss of its function. Okay, so actually how this disease is going to be inherited, it is inherited like autosomal recessive disorder, meaning that, because as you know, every gene in our body, we have 2 copies of that particular gene. One copy is coming from the father and the other copy is coming from the mother. So in case of cystic fibrosis and almost all cases of autosomal recessive disorders, if you have one copy is working normal and the other copy is not, then the clinical manifestations will not be there. And this case is called as a carrier. So there will be no clinical manifestation. But when we say that this is a patient, this is a case of cystic fibrosis with almost most of its presentations, this means that you have 2 copies of that defective gene. Otherwise, if you are a carrier, if you have only one normal and one abnormal, then you are a carrier. There is no clinical manifestations for the disease. So what are the clinical manifestations? Actually, the patient has thick mucus in the lungs, and because the mucus is a good medium for bacteria and for other pathogens, there will be repeated and severe chest infections. With the times, this patient with repeated chest infections will be ending up with respiratory failure. Also, the problem happens in the mucus that are present, mucus that's present, in the digestive enzyme producing organs and ducts that transfer them to the intestine. Mainly the pancreatic juice will be blocked from reaching the intestine. There will be defective digestion of the main foodstuff, mainly the lipid. So there will be malabsorption of main stuff or main kind of food, and mainly the lipids. So there will be steatorrhea, there will be weight loss with the time, and so on. Also, because there's a blockage of the digestive enzymes in the ducts that are transferring them from the pancreas, from the mainly exocrine part of the pancreas, to the lung, to the intestine. So there will be with the time exposure of the endocrine part of pancreas to damage. So there will be also future diabetes. Also, these patients will be liable for actually nasal polyps, liver diseases, male and female infertility, and osteoporosis as well. Now, we're going to talk about diagnosis of cystic fibrosis and then what's the role of PCR in this place. Actually, there are many diagnostic steps or many diagnostic types of this disease. First of all, it's screening. So this means that before marriage, if a couple is intending to marry and then one of them has a family history of cystic fibrosis, then they have to do the checkup. They have to know whether one of them or two of them carrying the mutant gene. Now, let's suppose that marriage has been there and there is a pregnancy. Then we want to know before the birth of the child whether this child is having cystic fibrosis or not. So this is what's known as the prenatal diagnosis actually. This is by examining the fetal cells while the fetus is still inside the uterus of the mother. So we have to take this kind of fetal cell and fetal DNA through one of these two things. Either first, amniocentesis, which means to aspirate the fluid that surrounds the baby within the uterus and to examine the fetal cells within the fluid, whether the DNA of the child is mutant or not. Second is by taking a piece of the chorionic villi by chorionic villus sampling, or let's say biopsy from the placenta, to see whether the fetal cell has the mutant DNA or not. Okay, so we are aiming actually to discover the presence of the mutation of CFTR gene in the fetal cells in this case. Now, as I said, the defect here in delta F508 mutation of the CFTR gene is that there is a loss of 3 nucleotides, loss of 3 bases, loss of 1 codon that codes for phenylalanine at position 508 of the whole amino acid sequence of the CFTR protein. So I expect that the defective allele or the mutant allele, and when I say allele, I mean the sequence of DNA, okay, the copy of that particular gene is going to be shorter than normal by 3 bases. If I can detect that by PCR, then I can prove the diagnosis, and I will show you how. Now, let's suppose that this is the normal CFTR gene, okay, and this is the abnormal or the mutant CFTR gene. As you can see there, this is shorter than the original one. There is loss here, right? So there is a shorter than normal, CFTR gene is shorter than normal CFTR gene in cases of cystic fibrosis. Okay, if this person, any person, if he has got the 2 copies are normal, then if you do amplification of the 2 copies followed by gel electrophoresis, then you can see only 1 band at position 63 base pairs. You will ask me, what does 63, what does 60 mean? This is by using a DNA ladder as a standard to know the size of the band. So if the patient or of that person, we have taken his DNA, amplified his DNA by PCR, and then we do this kind of electrophoresis, and then you can find only 1 band whose size is long, 63. This means that this subject is homozygous normal. This means that he has got 2 copies of normal gene. Okay, now if we amplify that particular DNA of that particular subject, and then we do the electrophoresis, and we can find only one band whose length is 60 only, this means that it's shorter than normal. This means that the person has inherited 2 mutant copies from his parents. 1 copy from the mother, 1 copy from the father, and both of them are mutant. So this is going to be called like homozygous affected, or a patient. He's going to have all clinical manifestations, as I said, respiratory, intestinal, etc., of the cystic fibrosis. But what if the patient DNA amplified by PCR and electrophoresis done, and we can find 2 different bands. One of them is long, like 63, the other one is short, which is 60. This means that this person has taken this normal band, normal gene from one of the parents and abnormal from the other. This is going to be like carrier. He's going to be carrier, called as heterozygote mutant or heterozygote normal. Heterozygote mutant, it means that this subject carries the abnormal gene in this case. So this is the aim of this lecture actually, is how we can use the PCR to diagnose a case of cystic fibrosis, example for inherited disorder. Of course, actually there is another kind of test which is used traditionally to test whether the subject has the cystic fibrosis, which is the sweat test. Actually, most of the time, because the parents, they kiss the child and so on, they can taste his salty skin, and they now suspect this case, so they refer to the doctor, and then sweat test will be done. There will be an increased amount of sodium monochloride in his sweat, and this is used actually more than the use of PCR because of the price, and also it's a kind of test which is known for a long time. Just simply we stimulate the sweating of the child, of the newborn, of the fetus, okay? So we just stimulate his sweating by certain kind of a drug, and then we collect the sweat on a filter paper or in certain device in a capillary, and then we examine or we measure the level of sodium nitrite. We can find that the salt concentration will be high. So, and at the end of this lecture, I hope that you have got the main points, and I wish you actually the success all the time, and thank you.